Hello, this is Bob Rutledge. In this presentation, I will describe some of the methods we have used to assess quantitative accuracy. One of the central attributes defining the performance of a quantitative assay is dynamic range. Historically, dynamic range has been determined by serially diluting a target in order to determine at what point quantitative accuracy is lost. This is an example of such an approach in which lambda genomic DNA has been diluted down to a predicted quantity of 2 molecules, with 3 technical replicate reactions conducted for each dilution. The tight clustering of the replicate profiles is reflective of the precision that can be achieved. An obvious exception is the 2-molecule sample, which I will return to in a moment. As was discussed in the optical calibration overview, LRE analysis generates similar values for delta E and Emax across this large range of target quantities. In addition, the precision of LRE analysis can be further illustrated by converting each respective F0 value into an optical calibration factor, which here produced a variance of plus or minus 16%. This indicates that quantitative accuracy can be maintained down to at least 19 target molecules. However, can it also be assumed that the extensive profile scattering produced by the 2-molecule sample is due to a loss in quantitative accuracy? This can be examined further by using the average OCF to generate predicted target quantities for each of the 3 profiles. This generates predicted quantities that range from 1 to 4 molecules, again suggesting loss of quantitative accuracy. Nevertheless, when these quantities were averaged, it predicts a quantity of 2.3 molecules, which is close to the actual target concentration. This suggests that scattering of these replicate profiles does not necessarily reflect a loss of quantitative accuracy. As will be illustrated over the next few slides, further investigation revealed that profile scattering is caused by Poisson distribution. This is an example of a low abundance transcript in which 8 replicate reactions were conducted, each containing 1 microliter of a reverse transcriptase reaction. Similar to the 2-molecule profiles presented in the last slide, this produced profiles scattered over a 2-cycle region. LRE quantification further predicts that the target quantities range from 1 to 6 molecules with an average of 4 molecules per microliter. How then does this relate to Poisson distribution? This graph was constructed using the Poisson distribution function within Microsoft Excel. Sometimes called the law of small numbers, Poisson distribution describes the probability that an aliquot will contain a specific number of target molecules when the target concentration is very low. For example, at a target concentration of 5 molecules per aliquot, only 17.5% of all aliquots will actually contain 5 target molecules. The remaining aliquots are predicted to contain target quantities ranging from roughly 2 to 8 molecules. Based on the fact that a 1-fold difference in target quantity will produce profiles separated by 1 cycle, a 4-fold range in target quantity is predicted to generate profiles that are scattered over a 2-cycle region. This is what was observed for the cDNA target presented in the last slide. This large range of target quantities generated by LRE is thus consistent with that predicted by Poisson distribution if the sample does indeed contain a target concentration of 4 molecules per aliquot. Importantly, Poisson distribution provides a simple approach to confirming the accuracy of this determination based on the prediction that as the target concentration falls below 1 molecule per aliquot, 2 events occur. First is the appearance of aliquots that do not contain any target molecules, denoted here in yellow. In addition, the frequency of these 0-molecule aliquots increases as the target concentration decreases. Assuming a lack of nonspecific amplification products, these 0-molecule aliquots will not produce an amplification profile. Second is the prediction that aliquots that do contain target molecules become progressively restricted in the number of molecules they contain as the target concentration decreases. For example, at a concentration of 0.5 molecules per aliquot, the vast majority of aliquots that do produce a profile will contain a single target molecule, with a small minority of aliquots containing 2 molecules. Thus, if this sample actually contains 4 target molecules per microliter, a 10 times dilution would reduce the target concentration to below 0.5 molecules. This in turn is predicted to produce replicate reactions that likely contain either 0 or 1 target molecule. And indeed, this is what is observed. As will be discussed in more detail shortly, the production of nil reactions is diagnostic of reaching what is called limiting dilution. However, the more obvious outcome of this analysis is restoration of profile clustering, which is predicted to be a result of amplifying single target molecules. If true, this would be a striking illustration of the precision that can be achieved with real-time PCR. Indeed, LRE quantification supports this contention, producing an average quantity of 0.9 target molecules. In addition, although I will not attempt to present more details, the tight clustering of single molecule profiles has been found to be characteristic of high-performance amplicons. Thus, in addition to verification of quantitative accuracy, this analysis can also be used to assess amplicon performance. Finally, before leaving this slide, I would like to present another method which I have called counting molecules. That is, if these are truly single molecule profiles, the average target concentration would be 0.5 molecules per microliter. This in turn predicts that the target concentration in the undiluted sample is 5 molecules per microliter, which is close to the quantity predicted by LRE. Nevertheless, Poisson distribution provides an even simpler and more reliable method for conducting absolute quantification. Wang and Spadoro developed an elegant approach to conducting absolute quantification based on the frequency of nil reactions. This approach, which I refer to as limiting dilution assay, provides a number of outstanding attributes. Foremost is that the analysis is based solely on the frequency of nil reactions. LDA is thus independent of the kinetic and optical parameters of real-time quantitative PCR. As such, LDA can be used to verify the accuracy of any quantitative PCR determination, irrespective of detection chemistry, enzymology, or instrumentation. LDA is also simple to conduct, requiring only that the target be diluted to about 0.5 molecules per aliquot, and that replicate reactions that produce nonspecific amplification products are either absent or can be identified. Let me then present two examples. This is a cDNA target with an LRE predicted quantity of 4,500 molecules per aliquot. The sample was then diluted 11,000 times and 16 replicate reactions conducted. This generated 10 nil reactions, which correspond to about 5,200 molecules based on LDA. Similar to the previous example, this produced 6 tightly clustered profiles that are supposedly produced by amplification of a single target molecule. Counting molecules supports this contention, predicting a target quantity of 4,100 molecules in the undiluted sample. Finally, LRE analysis of these 6 profiles generates an average quantity of 0.9 target molecules. In this second example, LRE predicted a target quantity of 11,600 molecules. Diluting the sample 22,000 times produced 11 nil reactions, from which LDA predicts a target quantity of 8,250 molecules. Closer examination of the profiles reveals 2 clusters, which are predicted to result from amplification of 1 and 2 molecules. This is supported by counting molecules, which predicts a target quantity of about 9,600 molecules in the undiluted sample. LRE quantification further supports this contention, predicting quantities of 1.4 and 2.3 molecules, respectively. Note also that the profiles are separated by precisely 1 cycle, which is consistent with a 1-fold difference in target quantity. This last dataset was taken from Figure 9 of our BMC Biotechnology article, in which 11 cDNA targets were quantified using both LRE and LDA. This provides additional evidence for the level of absolute accuracy that can be achieved, which for our cDNA targets is typically in the range of plus or minus 20 to 30%. To summarize, attempting to determine the dynamic range of quantitative PCR using serial dilution is confounded by Poisson distribution at target quantities below 10 molecules per aliquot. At such low concentrations, target quantification requires averaging of quantities derived from individual replicate reactions, which must also include zero-molecule reactions that fail to produce an amplification profile. The production of single-molecule profiles further demonstrated that LRE can maintain quantitative accuracy down to a single target molecule. Using an equation developed by Wang and Spadoro, absolute quantification can be conducted based on the frequency of nil reactions. Referred to as limiting dilution assay, or LDA, this method allows the accuracy of any quantitative PCR determination to be verified, irrespective of how the assay was implemented. LDA is technically simple to conduct and does not require a quantitative standard. LDA is also self-validating in that if the target is underdiluted, no nil reactions will be produced, whereas overdiluting the target will not produce any profiles. LDA thus provides a universally applicable method for independent verification of quantitative accuracy that in practice has proven to be exceptionally reliable. As always, thank you for your interest.